But you do see this.
We can see you, but we this slide is frozen. It does. I mean this. Oh, there you go. Okay. All right. So we see your PowerPoint, but if you
Yeah.
right.
But when I make it into a main screen, you just see blank.
Yeah.
All right. So maybe I can just do it the other way. Do it the normal way, like this, if that's okay with everybody.
Yeah, I think that should be fine.
All right. So one of the interesting things about this whole thing is that this family of materials basically allows us to basically have properties that are shear thinning, but also ranges greatly in what the properties are. So if you make these materials from 9% and you start changing the ratio of these two things, you basically can have a lot of variability in amount of force needed to make them into make them flow. So you can get materials that are very strong and you can get materials that are very weak. And all of these materials basically have the ability to, once you put them under the force, they become liquid-like, and once you remove the force, they go back to their original property. So this is the kind of stuff that we actually want with the material, because now you can actually inject it, and once it goes in its place, then it forms the right kind of features. Now, you know, we've done a lot of biological analysis and showing that it induces blood clot formation. So this example shows that what you can do is that you can have this material, you can have it in a way that it actually forms blood clots. So this is basically blood, a dish, and once you put blood, you see how long it takes to form the blood clot here. Typically, it takes a few minutes for blood clot to form. And even if you have gelatin without any nanoclay, it still takes a few minutes. Now, as you start adding this nanoclay, you get very rapid formation of the blood clot. So the time for blood clot formation with various concentrations of this material becomes much faster. The other thing that we can do is we can actually put it in different types of models, for example, seeing for its biocompatibility. So here, when we inject the material, we look at the biocompatibility over a few weeks. We see that the material doesn't have a huge amount of inflammatory behavior, and over time it degrades and it gets infiltrated with tissue. And this is a much more benign response than other types of hemostats that induce blood clot formation, like this commercial one, which you see even after 4 weeks has a lot of inflammatory response. We've done different types of animal models. You know, we've taken rodents and seen if the material works in their blood vessels. Here you can see the material inside their blood vessel, and you can see initially there's a lot of blood flow. Once we put it in the femoral artery, we block the blood flow completely. And we've done a lot of large animal experiments. And this is, again, the kind of work that we had to do to make this material attractive. And this shows that in these large animal experiments, you can actually block big blood vessels. This is original, it flows, and then here you can see the flow has stopped. And this here you can see the tip of the catheter and the material. And the material basically works in a lot of different conditions that we've applied. So, okay, so unfortunately here, I have to do these one by one. But when you do other models, like when you, this is initially the entire tissue is open. Once you start putting in your material, you can start decreasing this blood flow completely, and you can actually, again, induce embolization. So we've done a lot of other studies. I don't want to bore you with a lot of them, but we've seen that the material is biocompatible. So if you put it inside the blood vessels remain blocked over long periods of time, and it basically the material degrades and it's filled with infiltrating tissue. So it makes a fibrotic tissue response, which is the kind of response that we want long term. And many other experiments that shown its efficacy as well as its safety in different types of large animal models. So we did all this work, and then we wanted to actually translate this. Rami was super excited about this area, and he thought that it actually addressed one of his major clinical problems. So what we did is we started to think about forming a company which was aiming to redefine this therapeutic embolization area. So the first thing we had to do is to really find the right team. And a team that's really important, a team members that are really work well together are people who can communicate with each other, people who know their fields very well and are committed to the project. And we were very lucky to find Ehsan. Ehsan Jabarzadeh is also an academic entrepreneur. So he did his PhD in biomaterials. He understands the science, but also he did an MBA, so he understands the business side of it. So he partnered up with us, and he is very, very incredible person. I've been very, very lucky to team up with him. And Rami and I, of course, on the scientific side, were the engineer and clinician, and that's how we started off. Now the team has since grown significantly. We have a number of other advisors and other board members who are kind of involved now, but that's how we got started. And we actually had other starts with different team members, particularly different CEOs, which didn't work out as well. So that's really made me appreciate the importance of the right person who's going to champion the technology and push it forward. Really, all the work that Rami and I did was just a small part of what Ehsan has been trying to push forward. And now, over the past couple years, he's raised about $4 million in different types of seed rounds and different grants. And he's been really championing this, and it's just been wonderful to work with him. Now, what are the things that we had to learn through this process, which I think are going to be really important for everyone? So one is we had to really, beyond the science, dig into a lot of areas which were very uncomfortable initially for us. We had to understand the needs of the customers. In this case, the customers are the clinicians, but also their patients. They are all have a vested interest in the kinds of things that we do and what product is developed, and how we had to develop which market areas we're going to go over, because as I mentioned, this can be used in cancer, it can be used in embolization of aneurysms, it can be used in neuro applications. So there's a number of different things that one goes after. So we had to kind of define the opportunity and define the market. We had to look at the competitors, what other technologies were available, and what is their problems, what is the current standard of care, what do the hospitals like to use, what does it take for them to reimburse the process, who's going to pay for it. We had to look at our intellectual property portfolio and license these from the universities, and then the company has been building its own intellectual property portfolio as well. We had to look at a lot of things with regulatory, getting FDA approvals, the whole process of 510K versus a full pre-market approval, what they call PMA, and looking at the safety profile and the efficacy profile of our things. We had to do things that are even way, way beyond. We had to look at the production approach to make these in a GLP, GMP compatible process so that FDA actually accepts